So let me read what's written in our notes. So you can see the things like it's written that this HDL is formed from liver, GIT, and something known as you got an pre-beta HDL, you have got an A1, you got ApoC, ApoE, discoid, what is lacunin, all this is present. But now let us understand, take one step by step, let us understand it better, right? So if I show you the same structure here, right, if I show you the same structure of the metabolism of HDL, high density lipoprotein. So let us start with the first step. So normally if you see the structure of HDL, right, if this is a structure of HDL, you have got an apoprotein, right, you got a apoprotein 1, you have got a apoprotein C, apoprotein D, all this. But what happens here is, normally this is not present. Now it has to be synthesized. We have to produce the HDL, right? We have to synthesize the HDL. So this is the blood blood vessel where you can see this where it's going on in the blood vessel. All this reaction, what I'm going to tell now. So let us understand it clearly, right? So if you see this aspect, you have got a liver here, you got an GI tract, you can see the intestinal cells here. Right, you got an intestinal cell. Now the first thing is the main protein of HDL is ApoA1. If you see this, the structure of HDL, the main protein is ApoA1, and this ApoA1 is produced major source is by liver, and you can see it is also produced by the intestinal cells, right? You can see this, the red one, you can just appreciate this, and this is what the apoprotein is coming now, right? Right now the apoprotein, right, which is a protein of HDL, it is formed from the liver as a major source and is also from the intestine. And you can observe that we got one protein of whole of HDL, right? Now it starts getting all those other proteins, it starts recruiting, it starts collecting, actually starts collecting here. So once it starts collecting here, you can observe that it collects the other apoprotein like apoprotein E, apoprotein C, and you have gotten other enzymes also, other enzymes like LCAT, you have gotten CETP, you have gotten other substances also. And remember that this HDL contains all the apoprotein except the apoprotein B100 and apoprotein B48. And now if you see the structure of HDL here, it is looking flat, so we call it as discoid cell, right? It is known as discoid HDL. And observe carefully that why it is discoid HDL, because right now in the core you don't have triacyl glycerols or cholesterol esters, because it is newly synthesized, hence it is also known as nascent HDL. So we call it as discoid because of the flat shape, and we call it as nascent because it is produced newly. How does it produce? It is produced by the liver and the intestinal cells, where they produced ApoA1, and in the blood you have got other ApoE, ApoC, and you have got other apoproteins, and along with that you have got three enzymes. You got enzymes known as LCAT, you have got an cholesterol ester transfer protein, and you have got a lecithin we'll be discussing. And right now this is the structure of nascent HDL or discoid HDL, and this is what it's written in our notes. And if you observe carefully, this ApoA1, right, if you observe carefully, this ApoA1 has gotten positive action on this LCAT, on this enzyme LCAT. So it has gotten positive action. So now let us understand that, and this is part 1. Right now you have gotten nascent. Suppose I have already spoke in the last video, if you have seen the last video here, we already spoke about how the peripheral cells store this cholesterol, right, in the form of cholesterol esters, right? And suppose if you think this is a peripheral organ, say it is heart or muscle or any of the cells here, normally they store in the form of cholesterol esters, right? This is the peripheral tissue. Suppose if there is excess, right, if there is excess of cholesterol which it cannot store in the form of cholesterol esters, so from the peripheral cells it moves out into the blood, with the protein known, you have got a protein transporter known as ABC, right? ABC T transport, ABC A1 T transport. What is that? It is ATP binding cassette transport, and these transport are present on the peripheral tissue. So through these transport, you can see the cholesterol is coming out. So right now you got a cholesterol here, and right now you have got a nascent HDL. And let's see the action between these cholesterol and these nascent or discoid HDL. Right? If you observe the structure of the nascent here, all they are vacant, right? You can see this vacant, and we call it as posh word as lacune, right? Lacune is a vacant space. So right now we have got a lots of vacant space. Now it needs some cholesterol to be collected, right? It needs some cholesterol. You need cholesterol so that it can store in its core. So what happens is the nascent HDL slowly moves near the cells. Wherever the cell has got an excess cholesterol, it moves near the cell. So the moment it moves near the cell, you can see that cholesterol is coming out through this transporter. And remember that on this nascent HDL, or the discoid HDL, you got an enzyme. What is that enzyme? That enzyme is known as LCAT. What is that? It is LCAT. What is LCAT? LCAT is nothing but it is lecithin, C for cholesterol, A is acyl transferase, lecithin cholesterol acyl transferase enzyme. So what happens here is, if you observe carefully here, this red one LCAT takes the cholesterol inside, but how did you get cholesterol esters? And for the esterification of cholesterol, what is esterification? Esterification is nothing but COC bond. Now there is a bond between this, and let me tell you that along with the transport of the cholesterol, this LCAT has got an other function. The other function of the LCAT, if you observe carefully here, the lecithin, right? It converts the lecithin into lysolecithin, right? It converts lecithin into lysolecithin. What is the other name of lecithin? Other name of lecithin is phosphatidylcholine. And you can observe that the difference between the lecithin and the lysolecithin is, lysolecithin is without fatty acids. Lecithin has fatty acids, right? Lecithin has fatty acids, and after the conversion, this LCAT converts the lecithin which has fatty acids to lysolecithin which do not have fatty acid. Now where did the fatty acid go? Yes, the fatty acid had gone along with the cholesterol, and now it has become cholesterol esters. So right now it fills the space of a lacune, right? So this process continues. Actually this process continues, and now the situation has come. Right now, if you observe carefully here, we have got a lots of cholesterol esters, right? The same process continues. It takes all this cholesterol, extra cholesterol, and we call it as, right, what did we call this? Right now, since it has gotten cholesterol esters, we call it as HDL3. So this is what you have to understand. The meaning of HDL3 is because of intake of cholesterol in the formation of cholesterol esters, we call it as HDL3. Now let's see what happens to the further reaction. So same thing I am explaining whatever is present written in our notes. You can see the lecithin cholesterol transfer. You can see this you can see the lacune here. Right now we are here, right? LDL3. Now observe that I have already told in the last session that on these HDL you have gotten other receptors. The other receptors are cholesterol ester transport proteins, and those are the receptors which are present on the HDL. So let me draw this structure of HDL here. Yes, this is HDL3. You can see a lot of cholesterol esters here, right? You can see a lot of cholesterol ester, and these cholesterol esters, if you can observe carefully here, now we got a receptors. These receptors I have already told you, those are known as CETP. What are those? Cholesterol ester transport proteins. And now in the blood you have got an VLDL, you have got an LDL, you have got an IDL, right? IDL. Now these have got a receptors on these. They activate these CETP. They activate the cholesterol ester transferase protein. Once these VLDL, LDL, IDL which is present in the blood, once they activate these receptors, now we can see the exchange of substances, and you can see the exchange here. the moment it gets attached to this, you can see this HDL3 has got a lots of cholesterol esters. It has got a lots of cholesterol esters. So the cholesterol ester is donated to this. The cholesterol ester is donated to LDL. The cholesterol esters are donated to the VLDL. But VLDL, you can see that they are rich in triacylglyceride. So they give triacetylglycerol to this HDL, and now right now this HDL has got less cholesterol esters because it has donated to them, and right now it has got a lot of triacetylglycerol. So we call such substance, such HDL as HDL2, and this process again, once in right now what's going on in this aspect, you can see a lots of triacylglycerol here, triacetylglycerol, and you have got less cholesterol esters. So you have got a vacant space here, right? They have gotten less cholesterol esters. So what the process, again the process begin. How does the process begin? Again, once it goes near this peripheral tissue, again the cholesterol is released from the peripheral tissue, where they activate LCAT, and you can see influx of cholesterol inside, and you can see cholesterol esters are located here, and this process continued when the last cholesterol do not esterify. That means it is not bounded with fatty acids. So right now it is stopped. The intake of cholesterol is stopped because the last cholesterol, the entry of the last cholesterol is not esterified. What do you mean by esterification? Esterification means it is not bounded to the fatty acids from these lecithin, right? Now let us understand that what is the fate of these HDL2. Right, what is the fate of this HDL2? So let's see the fate of HDL2. You have got two fates of HDL2. One fate here is once the HDL go and interact with two receptors, and other fate is the HDL goes and interact with only one receptor. Now let us understand the fate of HDL. Suppose if this is an HDL, this HDL, right, HDL2 interact with these, right? What are those receptors? The two receptors are SRBI, right? What is SRBI? SRBI is scavenger receptor B1, SRB1, and you have got other receptor known as hepatic lipase, So this HDL slowly comes near the HDL and interact with these receptors. Suppose if it interacts, right now it is interacting with two receptors, right? So let's see, once it interacts with SRB1, what happens? You can see this. What happens? You can see this. You can see the cholesterol which is present in the HDL is taken inside the liver, and the cholesterol is taken right now under the influence of SRB1, that is scavenger receptor B1. This liver takes only the cholesterol ester inside, and with the interaction of this hepatic lipase, this liver takes triacetylglycerol, and you can see lipase breaks this glycerol, right? Breaks this triacetylglycerol and converts into fatty acids and glycerol. So right now the liver has took all the things, in the sense liver took cholesterol ester, it has took triacetylglycerol, so nothing is present in this core. So the moment it goes, right now it has donated everything and nothing is present, and it looks like a nascent or the initial or the precursor or the initial HDL. So the HDL which is formed now is nascent HDL. Either the HDL can donate everything and become a nascent HDL, or it can do the other thing also. This HDL, once it start interacting with this hepatic lipase, now the hepatic lipase, now it is acting only on the hepatic lipase enzyme, right? And this hepatic lipase takes the tag, that is triacetylglycerol, and converts into fatty acid and glycerol. So right now in the core you have got lots of cholesterol esters, and it's looking like HDL3, right? Because even in HDL3 we spoke in the this session, if you could have seen this HDl3, we have got lots of cholesterol esters. The same thing is present here. You have got a lots of cholesterol ester and it's looking like HDl3. So you have got a two fates here. Either it can interact and form a nascent, or either it can interact and form the HDl. So now let us read what's present here. Notes the extra points which is present in our notes. So if you observe the things here carefully, so we know that right, pre-beta HDL is synthesized in plasma by ApoA1 capturing. We read this, some of ApoA1 secreted in urine. So right now on this, when it takes all that, the liver takes all the substance like cholesterol esters and triacetylglycerol, it's not taking ApoA1. Apo is secreted in the plasma only. All Apo proteins goes to liver except A1, they enter into circulation. So I have told you that HDL2 has got a two paths. One path is interacting with the liver and liver takes everything like triacetylglycerol, and you have got an cholesterol esters. So it even takes all the apoproteins except the ApoA1, so that the ApoA1 lives in the circulation. So that is what it is saying. In the circulation it is excreted in urine. And you have to observe that the transporter of free cholesterol from cell to HDL, right? You have to understand that who are the substance, or who are the guys who is helping the transport of cholesterol. If this is a peripheral tissue, this is the cholesterol which is present here, and who is helping that helping or transporting the free cholesterol to the HDL. So let us see this free cholesterol to the HDL. So the first one is the receptor. I have already spoken. The receptor which was along here is ABC receptor. What is that? It is ATP binding cassette A1. So that helps in the movement of cholesterol from the peripheral tissue into the HDL. And next you have got ABCG1 also plays an important role from cholesterol to HDL. And you have got other substance, even SRB1, right? The SRB1 is present in hepatic, we already spoke that, right? And extra hepatic cells also. It has got dual role. What is a dual role? One dual role is it takes the free cholesterol to the lipoprotein one, and I have already spoke about that lipoprotein cholesterol to cell is esterified. So you have to understand that there you have got two, right? So the two substances I have told you here. One function of SRB1 is transport of free cholesterol from tissue to HDL, and next one is in hepatic cell. I have told you transport cholesterol esters from HDL to liver tissue. So I have told you this is the substance here, and this is, suppose if this is SRB1 and this is HDL, and HDL is the one which transfer the cholesterol esters into the liver. So this is all about the substance which is written in our notes regarding HDL. Hope you understood, guys. This is all for now. Hope I didn't miss any of the line here. I spoke about even this also, cholesterol esters TP. I spoke about the scavenger. These are the scavenger reactions between these LDL and these HDL. So this is all about the HDL, guys. Hope you understood. Let's meet in the next session. Thank you.